Hey everybody, what is going on? It's Alexander Williamson here with The Secret History Living in Your Aquarium. So, today we are going to be talking about something very interesting on episode number 89 of Fishtory with an exclamation point for style points. So, thank you to the Aquatic Morning Show, Jess, and all of you watching for making this possible. And an extra huge thank you to all of you members of the Secret History channel on YouTube or Patreon who allow me to pay for all the subscriptions to scientific journals. A lot of times just reading one article is $40. So to get you guys this info, to copy and paste it, to put the important parts into the document file that any of my members can see in the community tab, it really helps out to have your support, and I'm able to bring lots more news and do way better research, all thanks to you guys, so thank you. So, if you're watching as a member, you're going to breeze through all of these easily, and they're always audio-based, so you can listen to them as a podcast also. But if you're on the Aquatic Morning Show, I hope you guys are doing well and chatting in chat, and hopefully I'll be there right beside you. So, new research has been suggesting that plants are, in fact, heavily influenced by the microbial composition of aquaponic systems, which seems like a no-brainer to a lot of us, right? But researchers at the University of Gothenburg and at Wageningen University, Wageningen, I cannot even say it, and research grew lettuce or Lactusa sativa under a variety of hydroponic conditions, including nutrient supplementation, using different commercial hydroponic solutions, nutrients derived from the aquaculture of the Nile tilapia, Oreochromis niloticus, and then with the inoculation of also a probiotic or culture from soil. Remember, aquaculturing usually is using just the roots being immersed of plants in order to get them to produce fruits and vegetables and organic matter you can eat at a far faster rate by basically giving it steroids, by feeding it extra nutrient highway right to the roots, and requiring way less soil, way less water. And basically it's really revolutionized the way we can grow plants indoors, and it's revolutionizing the way we can have plants, period. I mean, this is how you get hothouse peppers in the northern parts of the United States in February or in November. So it's pretty amazing, and it's really changing the world. But one of the things that aquaponic systems have historically been doing is trying to sanitize their systems. This is where the aquarium news comes in and why this study is so important. So it turns out, as we've known for a long time, that there are different fungal relationships, bacterial relationships, and even little parasitical microorganisms that have relationships with different plants. A lot of those plants need those in order to fix nutrients or exchange carbon or sugar in exchange for N2 or nitrogen that they're able to access, because in our atmosphere they can't get the nitrogen out of it. That's what legumes are. You know, they have a partnership built in basically, and they have little nodes underground that store the nitrogen, so they're considered nitrogen fixers. Those plants are. Well, now in aquaponics and aquatic plants too, it's becoming very clear that the role of the microbial community is mediating fish and plant cultures, and it is considered the kind of holy grail of aquaponics. And despite widespread recognition that plants depend on their rhizosphere, or the zone where the chemical, biological, physical, fungal, bacterial, and everything else in the roots all interact, the extent to which this aquacultural zone occurs and what's in it is not well understood, and researchers have done hardly any research on it because of the diversity found. So this recent study actually highlights the importance of plants in determining their own rhizosphere. So that is to say, the composition in the soil that you'd usually find around these plants that helps them fix nutrients and things is present also in aquaponics. And a lot of times they assumed that it just came into the water, it was seeded, it was kind of like when you cycle your tank, it's just in the air, like different yeasts and spores from fungi are. But in this study, they controlled for all of that. They both tried inoculating it, they tried not inoculating it, they tried using a soil inoculation, so assuming that whatever it grew in water was the same as in hydroponic systems where the roots are underwater and in a substrate and the rest of the plant is above. And researchers conducted a taxonomic study, 16S RNA barcode study of the microbial communities originating from the water source, the hydroponic nutrient solutions, and the supplemented biofilter in the system. So they're trying to test for all the bacterias and different life forms that are colonizing surfaces in there, including those stockpiled on the surface and within the tilapia. And there are two primary sources of nutrients in aquaponic systems. There's the food for the fish and the fertilizers that fill in the gaps that the fish waste does not fill in. So the microbial inoculation can originate from an aqueous environment or from the air, like I said, just drifting in, or it can be introduced by actually putting spores of something or a piece of a colony of bacteria in the water, kind of like we do when we jumpstart filters. But recent publications have focused heavily on the diversity of these organisms in these systems, and that has given a rise to a hypothesis of how microbial communities can lead to an increase in yield and health of these plants. And they've found that chelating agents, cofactors, enzymes, and hormones that influence the bioavailability of these nutrients, or rather the plant's ability to consume and metabolize it, is being completely throttled or accelerated by these bacteria that live on the roots and these fungi that live on the roots. So it's becoming clear that the aqueous nutrient concentrations or plant fertilizers, but they call them aqueous nutrient concentrations in the study, play a more predictive role in determining what will be able to grow as a microbe community. And sterilization, which is common practice in aquaponics, actually becomes overridden by whatever's in the room. Whatever is around will make do. But they found essentially when they introduce a new bacteria and a new rhizome ecosystem that was in tandem with that plant in the wild or on farms in the past, that they're getting very, very different results than when it's left out, or than when they give it a different rhizome biome. So maybe they give it, maybe lettuce, let's try the biome that kale has, and they're finding that it drastically is impacting the diversity of the other things, the uptake of the nutrients, how much fungi and the mycelium and hyphae end up sucking up that nutrients and keeping it, versus how much they can use those nutrients, rearrange them, and sometimes more effectively give them to plants. And all of this is directly applicable to our own aquariums in trying to figure out if there are different bacteria, different fungi that can do that and basically feed our plants. It's basically like an IV, and it's a symbiotic relationship where they share nutrients, and it can help them grow far faster. So it may allow plants to be grown in regions and in environments, even within hydroponic or aeroponic environments, where in the past we haven't been able to get, say, potatoes or something to grow without having a full substrate layer in there. Well, now there may be ways to actually grow these things if you just inoculate the right bacteria and right fungi and right archaea and all those little microbiome factors. They may be able to allow us to do incredible things that we didn't think were possible for these plants and species and of course increase the yield or decrease the yield, decrease pests, decrease genetic problems, and also move us away from the whole monoculture thinking, because every one of these plants evolved for different partnerships, and it's going to need to be pretty intense research on every plant to figure out the most ideal pairing. But I just thought this was awesome news, and they're finally starting to do the DNA sequencing related to this subject, and I wanted to let you guys know that there is a new paper out about it that's got some interesting information, and it's Victor Lobanov and Carl J. Kiesman and Alyssa Joyce, and it just came out about 5 days ago, this paper, in the what's it called, the it's called Microbial Composition in Hydroponics and Aquaponics, as well as the Mycorrhizal Biome, and it was in the Frontiers of Microbiology journal. So I've linked that article down below, and you can Google it. But thank you guys so much for watching. I know this was a long one to start off. The next one will be extra short, just to make up for it. All right, I'll see you guys tomorrow. Bye. Hey everybody, what's going on? We are on episode, well, I can't really do it with one hand or two hands very easily, but we're on episode 90, so isn't that fun? Thanks for allowing this little series to become a mighty behemoth amongst all of those fish-related news web series that are out there. So today we're going to be talking about something that I know a lot of you are gearing up to put outside in your ponds right now, especially in the United States and Southeast Asia, and that is rice fish. Well, it turns out that rice fish, or there are several species, but usually it's the Japanese Medaka rice fish that we're talking about here, or the Oryza latips, and they're kind of a topwater killifish if you don't know what they are, but they are a lovely fish that can withstand freezes. They can withstand almost up to 100 degrees Fahrenheit. They can withstand low oxygen, high oxygen, low flow, high flow, and they have babies. They lay eggs by holding a clutch underneath their belly with their paired fins, and then they deposit them on eggs, and a week to 3 weeks later, depending on the temperature, there are babies from those eggs, and they're actually a really great fish for not eating a ton of their babies, generally speaking, most strains. Well, in Japan, this has been known for a long time, and they've been kept as both mosquito control type fish and just a lucky fish. It's even been incorporated into like Chinese and Taoist and Confucius, different home styling and design schools of thought, like feng shui and just various minimalist setups. And oftentimes it's seen as a good luck totem or good luck charm to put one of these bowls that can be very, very shallow and very, very small, yet support a breeding ecosystem of these little fish in your doorway, outside on the porch. And the UK is saying that not so fast. Their big governing body of pet imports and animal imports has said that they need to be instructing people that these are indoor fish only. No tubs, no backyard ponds, none of that. And if they're seen outside, one, they're now starting to create regulations and punitive measures like, tickets and things, or confiscating the fish if they are found to be outside, even on your property, or on your roof even. But it does bring up a really good point, which is when we hear legislation like this in any country, any Western country, or any country that's even remotely similar to the United States and has an influence on the United States, we as fish hobbyists need to be stepping up. This is how things like the Lacey Act get, the amendments to it, get written in and just go crazy overboard and say, let's ban everything except for whatever we specifically list as not banned. And rice fish are going to be one of those top priorities, most likely, because they can live in almost any environment. on Earth other than the Arctic or, tundra, polar regions, and maybe high alpine. Although there's even reports of that, like in the Alps. There's reports of them surviving there, and in the mountains, in areas like in the Teton mountain range. But regardless of all of that, it's just a reminder that we need to be responsible with our fish, and it's not so much keeping them in tubs and things. There is the potential that birds would land and then take a piece of hornwort or some duckweed that has an egg or a few eggs stuck onto it as they do release those eggs every day, and they kind of stick them to things like a rainbow fish would, just like you would see with fish that use spawning mops. Well, then birds come into the water trying to eat the fish, and oftentimes, inadvertently, they may eat a fish, or try to, and then they fly off with a bunch of eggs on their leg that they drop off in the next body of water. So it's a reminder of the possible implications of if you are allowing any waterfowl or raccoons or any creature that could possibly get plant material on them, this is how things spread. This is how bluegills, bass, stickleback, perch, this is how a lot of them have spread in foreign or alien environments, and why they are now present in every state in the United States, even if they were introduced as game fish into one region originally. So I just wanted to put that out there as a reminder to, if we are doing these things, let's think about it for one, and think about how you publicize it. It's something that I've never done in the past, and maybe it's something that I should more low-key, show myself building a setup indoors, something like that, because I could easily see this being adopted in the United States, regardless of what happens with the Lacey Amendment changes, but definitely in Canada as well, where meteor minnows are not allowed in many places, or white cloud minnows are not allowed in many places, and same with a lot of European countries. So keep your eye out, and let's just be sensible, guys. Just a bit of a warning and a little bit of news out of the UK that everyone got a big warning. All of the importers and transshippers were told, like, this needs to be said to everybody you sell it to, that these are indoor fish only, because they just spread too easily. And it's something to think about for our own ecology here in the United States. Granted, we already have those bass and perch and other fish pretty much in every body of water that these fish would make it in, so it's not quite as likely that they would survive, according to a few ecologists who weighed in on the topic in the comments. But I just thought I'd bring that up. So that's the news out of the UK. Banning more things, I suppose. Alright guys, I'll talk to you next time. Bye. Hey everybody, it's episode 91 of Fishtory, where we go over new discoveries, new stories, and new just fun info about fish that comes up out of academia, out of the news, and out of the hobby every week. And we try to hit it 4 days a week, and then we have a couple live streams on my channel, a couple other videos. Pretty much there's news every day about the amazing things going on in the hobby and in the science of freshwater and saltwater ecology and fish. Well, today we're going to be talking about how the electric catfish, the electric eel, how mormyrids, any catfish or bottom fish or eel that uses electric signaling, we're going to be talking about how they evolved, because a recent paper has figured out the mystery of how in the heck does an animal just suddenly become able to shock people or send out signals, even if they're weak signals, like mormyrids or like electric eels. So this is basically a analysis of their genetics and comparing all the species around the world that are able to produce electrical signals beyond, there was a certain threshold cut off, and I don't know a lot about electricity, honestly, so I don't know in those low units what that means. We're talking, organic amounts of electricity, and everything puts off a biomagnetic field, and everything creates a little bit of static electricity and friction, so I don't know all that. If you want to know more of that, you can read the paper yourself. But electric organs help electric fish, and everything from the electric eel to the marmorids. They're pretty amazing. They send and receive signals akin to language or bird songs, and they're able to recognize the other fish in their species, sometimes even in their genus. They can figure out the sex, the health, and what they're up to, if they're feeding, if they're scared, and much, much more by these electrical signals. So it's thought that these electrical signals probably evolved as a way to communicate first, not as a way to hunt or shock or hurt things, because it probably happened very slowly. So it probably was a current that maybe when they got really excited, they'd tense up these special muscles, and those cells in those muscles would respond by putting off a signal, and maybe that signal could be detected by other fish in the water. But the whole how they perceive these signals, that is a whole different paper and can of worms to unpack. So let's just talk about how now they have essentially they think they have figured out how it evolves. And scientists were trying to pinpoint the exact gene and mutation behind how this works, which would also unlock a lot of diseases and how they work in humans as well. Things like restless leg syndrome, or they have another one called exploding head syndrome, which is like loud noises in the middle of the night. As well as just nerve pain and neuropathy. They want to know what it is that's going on that has changed their actual nerve cells or muscle tissue into this all new arrangement. And all fish have duplicate versions of the same gene that produces these tiny little muscles. And in the tiny little sections of muscles, they have tiny little motors, and they're called sodium channels. So to evolve electric organs, electric fish turned off their sodium channel gene in those little muscles and turned it on in other cells. So the tiny motors that typically would make muscles contract and basically tell them with a signal of electricity to suddenly tense up or release, and usually when they release, they don't have the electricity going through them. They found a way to basically, genetically, what happened was two evolutionary steps, which first the cells that are being used for normal muscle function had to slowly fade away, and at the same time you also had to have a different mutation where these other cells are taking the same little motor that flexes a strip of muscle, and they're taking those cells like the ones in the heart that makes your heart go dum 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 dum. Well, instead of that, it's making a little friction or a little spark, basically, not a real spark, but a little electrical signal, and storing that up in hundreds of thousands of cells arranged in kind of an order. So it's almost like a battery with things like sodium and lactic acid and ATP and all the different things that cause energy in fish. They're able to metabolize those things, and then at the cellular level create an actual electrical pulse or signal, which is pretty cool. So this is exciting because we can see how a small change in a gene can completely change where it's expressed. And in a study done by Harold Zacon, professor of neuroscience and integrative biology at the University of Texas in Austin, he was corresponding with another author of a paper from the same university working on electric signaling at Austin, and then they teamed up with some folks at Michigan State University and described discovering a short section of the sodium channel gene, about 20 letters in the GTAC letter chains, the four different possible characters that make up our DNA, and it controls whether the gene is expressed in a given cell. So they then confirmed that that is what is being expressed in all electrical fish, whether they're in Africa or South America or whether they just have, like in some catfish, a heightened sensitivity to being able to sense the conductivity of water and things. They also found them present there. So now they're wondering which fish have them that we may not even have any idea have them, and our equipment might just not be tuned fine enough to hear them. So that's why these little sodium channels are so important, is because first they have to be told to turn off, then another gene has to be told to turn on and put this altered version of that back into its place. So it's basically one cell being told don't replicate, and then another one of a completely different form, which are these little ion producing cells. They're able then to kind of plug into that same thing and use the energy and the quote unquote snap or contraction of the muscle for quick muscle activity to then produce electricity. And when in a line, they're able to amplify that over the length of the entire muscle and tissue there. So Zacon said, so the next step in terms of human health would be examine the region in the database for humans, because we have these same genes, and to see how much variation there is in normal people, whether some deletions or mutations in this region could be leading to lower expressions of sodium channel signaling, things like restless leg and neuropathy, and what we can do about also telling those same sections of those cells to turn back on and regain functionality, which is really cool. Really, really neat stuff. So I thought this was a cool story, and there's a whole lot more to read about it. But Zacon said that the sodium channel gene had to be turned off in the muscle before any sort of electric organ or cell array could be evolved. And so they probably had those cells somewhere in the body, and then they decided, just by chance, it was decided by fate that was turned off. The muscle signaling was turned off, and an electric organ had already been forming, and then by chance was placed right there with those cells that were not firing. So you may have had a very slow fish in these interim processes, like an ambush predator that lives in the mud, which makes sense with catfish and eels and the fish that we know that do have strong electrical capabilities. So with all that, the new stuff that was happening to the sodium channels in the electric organ would also be occurring in the muscle. So it was important to isolate the expression of the gene to the electric organ also, and where it could have evolved without harming the muscle tissue, without shocking the muscle tissue. So there are two groups of electric fish in the world, the one in Africa and the other in South America, and researchers discovered that the electric fish in Africa had mutations in the same region, while electric fish in South America had actually lost it entirely. They'd evolved past that, and they didn't have it turned off. They were just missing those gene cues. But both groups arrived at the same solution by developing an electric organ and losing expression of their sodium channel gene in those organs, or in that muscle, whatever you want to call it, while it's evolving. And you have two different pathways and two different systems, and that's why the New World fish are so much better at shocking and actually damaging or harming an animal, is because they likely evolved it much earlier, but on separate continents. We're talking millions of years apart. So pretty cool stuff. And if you're into all of this, there are two great papers that just recently came out in the last year and a half or so, plus another summary of those papers kind of linking their work together. And it's kind of a mystery too, like how long this took or what other jumps were involved. But now they've singled out like what happened, when it happened, but they just don't know how long or how many cells had to get in a line randomly to become an organ, so to speak, or when does tissue become an organ that's functional. So I know that was a lot, but I thought it was super interesting, and the National Institute of Health is funding this research, so it really applies to humans, even like Parkinson's and things like shaking muscles or Lewy body dementia. They're looking into if there are possible epigenetic or maybe CRISPR type insertions that could be made someday in the future that would literally shock the muscles into doing that, or tell the muscles to stop doing that, or tell the muscles to become some other type of tissue, half and half. The sky's the limit, so it's pretty interesting. All right, guys, thanks for sticking with me on this long one, and I will talk to you next time. Everybody, what's going on? It is the last episode of the week, and we have a mystery. We have a mystery for episode 92 of Fishtory, and that is that at the Shed Aquarium in Chicago, which is the oldest aquarium in the United States, it's like 160 years old and still operating. They have a mystery. So during their quarantine process, all their animals receive antibiotics, a dewormer, and an anti-parasitical. And this is when they catch wild animals out in the sea and then bring them in. Before they go into the community tanks on display, they go into a quarantine tank and they receive a common anti-parasitical medicine, which is chloroquine phosphate. Now, you might remember chloroquine, hydroxychloroquine, the fact that chloroquine was in a whole bunch of koi pond anti-parasitic medicine, and people drank it in certain cases, and over half a dozen people died during the whole viral outbreak we had the last 2 years. It's probably way more than that, but those are the for sure cases, like bottle in hand. They were alive, called in, and were like, whoops, I messed up, now I'm sick, and we weren't able to do anything. So obviously we knew that stuff could be harmful, but in any case, we have to dose it in low amounts to get all the little parasites and critters off of things like sharks or stingrays or gobies or little reef fish, whatever it may be. They all need it. So they basically dose the water column in this quarantine tank. So this pharmaceutical bath that they have, it's kind of a trio really, has been established for about 30 years. And they've been adding chloroquine to the water more recently, rather than flubendazole or fembendazole, just because they felt it was easier on the fish, and also I guess it was cheaper too by a little bit. But some aquariums have a different procedure, so they need to maintain a certain amount of concentration of this stuff in the water for it to actually kill the parasites. But soon they started noticing, about 2 years ago, that the chloroquine was going missing. So they would dose the correct amount, a day later come back and check it, and it'd be 20% lower than that amount they put in. Well, the next day they'd check it again, and it would be even lower. And by the end of the week, it would be completely absent, when normally it should be sticking around that whole time until they use a charcoal filter, pull it out. And, you know, some of it deteriorates over time, a small margin of it, but most sticks around. So aquarists started collecting water samples and swabs of every surface they could think of, both in the filtration systems behind the scenes, in the enclosure, on the glass, on the hardscape, and even on the fish, and trying to figure out what was going on. So they then ran all these little swipes, and basically they were looking for if there was something there, either chemically that was able to break things down that something was producing. For instance, if there's coral in the quarantine tank, or some basic, reef element, maybe there was an animal that was new that they didn't notice, some sort of little creature that had a byproduct that was interacting oddly with the medication and causing it to be destroyed. Because the other medications, the antibiotics and the anti-worming or the deworming medication, were still present and not gone at all, and nor were any of the water parameter fertilizing and phytoplankton and all the different things that they put in there for the corals and for the plants, fish, you name it, in both fresh and salt water. But this tank that it first happened in was in salt water. And after they swabbed the whole thing down, they got 754 different microbes. So to help solve this, they sent it off to the Northwestern University microbiologist lab, and they started to send in water samples and all those swabs to track down what was going on. So after concluding that microbial and chemical analysis of the samples from the saltwater aquarium must be the culprit, they weren't sure which of the 750 plus were the ones that were doing it. So they started to look and test each one and basically give it very, very diluted hydroxychloroquine in water, and the chloroquine sulfate, and a host of other things that chloroquine phosphorus also. So the things that most bacteria, most life in general needs at some cellular level is carbon, nitrogen, oxygen, and phosphorus. Those are the big ones. Those are the big building blocks. And they're basic necessities. Everything needs it, from bacteria to birds to fungi to plants to us. And Erica Hartman, who was in the Northwestern lab when this happened, decided to conduct an official study and research of this case of the missing chloroquine phosphate. Sorry guys, so many things with chloroquine in it in this story. But it looks like some variety of the microbes were actually using the medicine as a source of nitrogen, because when they looked at it after a few days under a group of 21 different microbes, they found that the molecule with the nitrogen part of the segment on it was completely gone. And I just thought that was bonkers. So the solution was that something evolved or already had the ability, and these hungry microbes were responsible for stealing the nitrogen straight off the molecule of this medicine. Just really, really fascinating stuff. And there's a full write-up of it in the Biology of Plants and Animals, or Biology of Cells and Microbiology journals. So it came out in 2 different journals, but most recently it says hungry microbes responsible for stealing Shed Aquarium's medication. And the Shed Aquarium released a press release about this, which has quite a bit of information on it too. But really, the scientific studies out of Northwestern had better info on it and a lot more details. And the thing is, this was saltwater, but they knew about other examples. They just mention it in passing of it happening in freshwater to carbon or to nitrogen or to oxygen that is in another compound that they're adding to the tank. So the question is, even in freshwater, or for sure in saltwater aquaria, are these native? I mean, we have a bunch of bacteria that, of course, it's there just to take ammonia, turn it into nitrates or nitrites, and then into nitrates, and then use that. Or also we have other bacteria and other processes that can do the reverse, or that can take N2 in plants, just like we were talking about earlier with legumes and nitrogen fixers, can take it out of the atmosphere, pull it down into the plant. Well, if that bacteria is growing on the roots of the plant, maybe it's stripping the nitrogen right off of those nodes, and maybe there's a relationship with a fungi or a microbe, but we just don't know yet. They didn't track it down any more specific than they knew it was in a genus with these 21 species of nitrifying bacteria, or rather nitrogen-eating bacteria, nitrogen-consuming bacteria. And that happens to be a big part of their filtration. So the overall thought was either completely gut the aquarium system, scrub it down with bleach, and get new pipes, new filters, new hardscape, everything for this big old tank, which is not a little tank. It's for large fish, sharks and things too even. It's not just for small community fish like we have in the hobby. But yeah, they realized that they tried to clean it, they tried to scrub it down, nothing worked. So they either are going to have to replace everything or just switch to a different chemical. So it looks like they might be going back to fenbendazole, flubendazole, but they're going to have to dose in massive amounts because it's a big, enclosure that these animals are swimming in, and they have to quarantine them for quite some time. But if you want to read more about it, I've got a link to the Northwestern School's McCormick School of Engineering and Microbiology and Physics, and they have more information on this whole shed aquarium quarantine tank chemistry and medicine disappearance act. But I just thought it was really cool, really interesting, and it's amazing what will happen when nature just decides, when nature steps in and just decides to find a way to survive. This could be a brand new strain or a mutation, and that's why it's suddenly doing it. Or it could be something that was there all along, but all of a sudden it realized, oh, there's easy food in this water. I can break that piece of nitrogen off easily. And yeah, I just thought this was super interesting. So the chloroquine phosphate is no longer probably going to be the go-to medication, and they're also trying to not cross-contaminate anything into any of the other tanks, but let alone any other facilities, any other aquariums. So thanks so much for making it to the end of the week. I know we had some long episodes today, some long-winded ones, but I hope you enjoyed them. Thank you so much for watching. It's Alex with Fishtory on The Secret History Living in Your Aquarium if you're a member, or on the Aquatic Morning Show if you're not. Either way, love to see you. Great to have you watching, and I hope you have an excellent week. And back to you, Jess. Goodbye.